In the second video on gene therapy, we're going to go over two specific examples of diseases that are currently being treated with gene therapy. First, I want to remind you of our learning objectives for these videos. By the end of this two-part video sequence, you should be able to define gene therapy, explain the considerations in deciding whether a disease can be treated with gene therapy, explain the difference between in vivo and ex vivo gene therapy, and understand why some diseases are better treated with one approach than the other. You should be able to explain the differences between lentiviral vectors, AAV vectors, and gene editing, and discuss the pros and cons associated with each approach. And finally, you should be able to design a hypothetical gene therapy strategy for a human inherited disease. The two examples of successful gene therapy treatments that we're going to talk about today are first, gene therapy to treat ADA-SCID, and second, gene therapy to treat Leber's congenital amaurosis. So first, let's talk about ADA-SCID. Before we get started, I just want to mention that a lot of the work on ADA-SCID was actually done here at UCLA in the lab of Dr. Don Cohn. So what is ADA-SCID? ADA-SCID stands for adenosine deaminase severe combined immunodeficiency. This is a very rare recessive disease affecting 1 in about 200,000 to 1 in 1 million live births. This is often called bubble baby disease because children with the disease are severely immunocompromised, and any encounter with germs can be fatal for these children. So as a result, the children were often kept in a sterile bubble to prevent them from getting infections. So what causes ADA-SCID? ADA-SCID results from loss of the ADA gene. And ADA, or adenosine deaminase, is an enzyme that breaks down adenosine and deoxyadenosine. Without ADA, deoxyadenosine builds up to levels that are toxic to cells, and particularly they're toxic to thymocytes, the cells from which T cells are derived. The result is that in the absence of ADA, thymocytes die, and then this results in a loss of T cells. As a result, patients are severely immunocompromised, and if left untreated, they often die within the first year of life. Now let's think about the factors that need to be considered when deciding whether ADA-SCID is a good candidate for gene therapy. The first question is whether it's caused by a mutation in a single gene, since these are the diseases that can most easily be treated with gene therapy. As I just mentioned, ADA-SCID is in fact caused by a mutation in a single gene, or mutations that affect a single gene, and that's the ADA gene. The next question is how complex the disease-causing mutation is. Loss of function mutations can be treated using gene addition, which is the most common and most well-established form of gene therapy. However, as we talked about in the last lecture, gain-of-function mutations cannot be treated by gene addition because the mutant protein will still be present. Now, in the case of ADA-SCID, the disease is caused by loss-of-function mutations in the ADA gene. So ADA-SCID is a good candidate for gene addition. And then finally, are there other treatment options available? In the case of ADA-SCID, the preferred treatment is actually hematopoietic stem cell transplantation if there is a matched sibling donor who can donate bone marrow. If there's a matched sibling donor, you can then reconstitute the patient's immune system from the transplanted stem cells. However, for this to work, the patient has to have a matched sibling donor, or the stem cells will be rejected by the patient's immune system. For patients who do not have a sibling donor, the disease is often fatal. So patients without sibling donors may be good candidates for gene therapy. And a big advantage of gene therapy here is that the patient's own cells are used. And so since we're using the patient's own cells, the cells are not rejected by the patient's immune system. The next question I want you to think about is whether in vivo or ex vivo therapy is most appropriate for ADA-SCID. So just to remind you, with in vivo therapy, genes are transferred into cells while the cells are still in the patient. Whereas with ex vivo therapy, the cells are taken from the patient, the gene is added to the cells in the lab, and then the cells are transferred back into the patient. ADA-SCID primarily affects T cells. And so treatment requires adding the ADA gene to hematopoietic stem cells, which will then give rise to T cells and other immune cells. Hematopoietic stem cells can be removed from the patient, treated, and then transferred back to the patient. So this is a form of ex vivo gene therapy. The next question is how the wild-type ADA gene can be introduced into hematopoietic stem cells. Should you use an adeno-associated viral vector, a lentiviral vector, or a form of gene editing? In this case, the ADA gene needs to be added to hematopoietic stem cells, which are dividing cells. And as I mentioned in the last video, AAV vectors do not integrate into the genome, and so they can't be used in dividing cells, because when the cell divides, they will not get passed on to the daughter cells. Lentiviral vectors do integrate, so after they're introduced into stem cells, they'll continue to be expressed in all of the cells that are derived from those stem cells. So in this case, it's necessary to use a lentiviral vector. Although I should mention that there are other types of retroviral vectors that can be used, but lentiviral vectors are the ones most commonly used today. Another possibility would be to use gene editing, but gene editing is not necessary in this case since the mutations in the ADA gene are loss-of-function mutations. Okay, now let's go over the general approach used to correct ADA SCID. First, stem cells are removed from the patient. Then a lentiviral vector expressing the ADA gene is delivered into the extracted stem cells. The patient is then given chemotherapy to remove their existing blood cells. And then finally, the stem cells, which now contain a working ADA gene, are returned to the patient. The patient's blood cells are then repopulated with cells derived from the modified stem cells. This therapy has been incredibly successful for curing infants with ADA SCID. As I mentioned, a lot of this work was done by Don Cohn's lab here at UCLA. And this UCLA newsroom story actually features one of the patients Dr. Cohn successfully treated. This is a family with 2 twin girls, and the one on the right was born with ADA SCID. So as infants, the girls had to be kept isolated so that the one with ADA SCID wouldn't develop an infection. But now, after gene therapy, she appears to be completely cured. So gene therapy has been hugely successful in curing ADA SCID. Okay, the second example we're going to talk about is a very different disease called Leber's congenital amaurosis. This is a retinal disease that can result from loss of a protein called RPE65. RPE65 functions in the retina and is part of the visual cycle. And it's important for converting all trans retinal back into all cis retinal. You don't need to know any of the details of RPE65 function. The important thing is just that as a result of the visual cycle being disrupted, patients lacking RPE65 have very severe vision loss. This is a rare recessive disease that affects about 1 in 40,000 newborns. And as I mentioned, it results in severe vision loss and eventual blindness. Now let's talk about the considerations in determining whether Leber's congenital amaurosis is a good candidate for gene therapy. The first question is whether it is caused by a mutation in a single gene. Well, the disease can actually be caused by a number of different mutations. But some of the cases result from loss of RPE65. So these cases where the disease results from loss of RPE65 are the ones that can be treated with gene therapy. The second question is how complex the disease-causing mutation is. In this case, the disease results from a loss-of-function mutation in RPE65, so it should be possible to treat it using gene addition. Finally, we need to consider whether other treatment options are available. And the answer here is simple. There are basically no other treatment options available for this disease. Now let's think about whether in vivo or ex vivo therapy is most appropriate for Leber's congenital amaurosis. In this case, the disease affects the retinal cells. And unlike bone marrow cells or blood cells, retinal cells cannot be removed from the eye and then put back in. So in this case, in vivo gene therapy is required. The next question is how the RPE65 gene can be introduced into the retinal cells. Well, the RPE65 gene needs to be added to retinal cells, which are non-dividing cells. So an AAV vector can be used in this case to introduce RPE65 into the retinal cells. By using an AAV vector, you avoid the risks associated with integration of viral vectors. So an AAV vector would make the most sense in this case. The approach here is pretty straightforward. Basically, you just inject the AAV vector containing the RPE65 gene into the back of the eye. And then it will infect retinal cells, and the infected retinal cells will start to express RPE65. This treatment was recently approved by the FDA and is being marketed by the company Spark Therapeutics under the name Luxturna. So it's one of the first FDA-approved gene therapies. So far, many patients who have received Luxturna have shown improved vision following treatment. And you can see one of these patients here. This is a boy who before had very severe vision loss, and now he can see well enough to fish and to read facial expressions. However, it is important to note that unlike the case with ADA SCID, where the treated children appear to be cured, in this case the therapy is not a cure. The patient's vision is improved, but it's not fully restored. It's also important to note that the long-term efficacy of Luxturna treatment remains unclear. In some clinical trials, the improvement in vision was only temporary, whereas in other trials it appeared to be longer lasting. So at this point, we don't really know whether these patients will continue to show improved vision in the long run, or whether ultimately their vision will deteriorate again. Finally, another important point is that gene therapy is incredibly expensive. It currently costs $850,000 for a single Luxturna treatment. And so because of this, there are obviously a lot of medical and ethical issues that need to be worked out in terms of making sure that the therapy is accessible to everyone who needs it. To conclude, now that you've watched this two-part video sequence, you should be able to define gene therapy, explain the considerations in deciding whether a disease can be treated with gene therapy, explain the difference between in vivo and ex vivo gene therapy, explain the difference between lentiviral vectors, AAV vectors, and gene editing. And finally, you should be able to design a hypothetical gene therapy strategy for a human inherited disease.